Hello students, in today's class we are going to learn about toxins. Bacteria that cause diseases can be of two categories. First are the infections. So these result from the growth and multiplication of the pathogen that leads to tissue damage or alterations. And the second is intoxication. These are basically resulting from the entry of preformed toxins into the host body. And toxicity is defined as the ability of an organism to cause disease by means of preformed toxins that inhibit host cell function or kill the host cells. So toxins can be broadly divided into two categories, exotoxins and endotoxins. So exotoxins, as the name suggests, these are extras like secreted out of the bacterial cells. They are soluble heat labile proteins. They are released into the surrounding as the bacterial pathogen grow, and the toxin may travel from the site of infection to other body parts, tissues, or cells, exerting its effect. The second type of toxins are endotoxins, which are actually part of a bacterial outer membrane that functions as a toxin. Let us discuss about properties of exotoxins first. So they are synthesized by specific bacteria that have plasmids or a prophage integrated within the genome that will encode for the gene for the toxin. Their heat labile means they will get inactivated at temperature around 60 to 80 degrees Celsius. They are highly toxic. So even small doses, about micrograms per kilogram amount, is enough to induce a toxic effect. And they are associated with a specific disease and have a specific mode of action. They're also highly immunogenic in nature, and they will stimulate the immune system and production of antibodies or antitoxins. They are also easily inactivated by formaldehyde, iodine, and other chemicals. They are also often named after the disease they produce. For example, diphtheria toxin that is involved in diphtheria. So exotoxins can be broadly categorized into four types. The first category is a AB toxin, which is made up of two subunits, the A subunit and the B subunit. The B subunit or the component is basically binding to the host cell surface receptors, and thus it helps the A subunit to enter inside the cytoplasmic membrane. Once inside the membrane, it will act and it will cause its effect. So A part is actually you can say the enzymatic part, while B part is helping entry of the A part into the cell. The second type of toxins or category is specific host site toxins. So these also might be AB type of a toxin, but it basically consists of those toxins that will affect a specific host site only. Then the third type of a toxin, they do not contain a separable A and a B part. And some of the examples of these includes hemolysins, leukocidins, and these act by disorganizing the host cell membrane. And the fourth or the last category are also known as superantigens, since they will activate or stimulate the immune system uh, and allowing by release of certain cytokines. So the first category, that is AB toxin, as already discussed, it consists of two subunits, the A and the B subunit. The A subunit is the enzymatic subunit which exerts a toxic effect, and the B subunit, also known as the binding subunit or the binding fraction, is responsible for binding to the target cell and allowing receptor mediated endocytosis mechanism for the entry of the toxin inside the cell. So just the isolated A subunit lacks binding, and hence it will not enter the cell. So it will be not showing any effect. Similarly, isolated B subunit only will lack the enzymatic activity, and hence it is non-toxic to the target cell. So let us discuss an example of a diphtheria toxin, which is an example of AB type of a toxin. So rats and mice are relatively resistant to diphtheria toxin. However, they show their toxic effect in humans, rabbits, guinea pigs, and birds. It is secreted by the organism Corynebacterium diphtheriae. As discussed, the B fragment will bind to the host cell surface receptor and will allow it entry of the A subunit inside the cytosol. Um, Later on, proteolytic cleavage will take place between A and the B subunit so that the A subunit will separate out and will be active in the uh, cell cytosol. So here, A subunit acts by disrupting the protein synthesis process by blocking one of the elongation factors, which is known as elongation factor 2, that is involved in the growth of polypeptide chain. So also diagrammatically on the next slide, I'll explain how the diphtheria toxin acts. So it is encoded by a tox gene, which is present in lysogenic bacteriophage, which is known as phage beta. So this process is known as phage conversion of Corynebacterium diphtheriae. So these are basically those bacteria which would be otherwise non-toxic and or will not be producing any toxin, but the toxin producing ability is because of the tox gene that is present in pathogenic strains of Corynebacterium diphtheriae, which are basically converted because of integration of the beta phage into the genome. So this image explains the mode of action of diphtheria toxin. So it is a AB type of a toxin. So B part will bind to the cell surface receptors and it will allow the entry of the A part. When entering the A and the B there will be a proteolytic cleavage and only the A part will get separated out from the B part and will enter inside the cell. So elongation factor 2 is an important growth factor. It is an important elongation factor that is required during the protein synthesis process. So it interacts with the transfer RNA and helps in the elongation step. However, because of the presence of the toxin, it will not allow binding or entry of the transfer RNA, as a result of which the protein synthesis process fails. Now, the second category of proteins are the specific host cell exotoxins. So these are categorized further on the basis of the site they affect. For example, neurotoxins will affect the nervous tissue. For example, the botulinum toxin or the tetanus toxin. Enterotoxins will affect the intestinal tissue, while cytotoxin will affect the general tissue. So diphtheria toxin is an example of cytotoxin as well. So neurotoxins are basically those toxins that will affect the nervous tissue. So tetanus and botulinum toxins that are produced by Clostridium tetani and Clostridium botulinum are important examples of neurotoxins. So these are normally soil microorganisms and they can occasionally cause disease. Clostridium botulinum, they will rarely grow directly on the body, but it will grow in improperly preserved food and thus the toxins are synthesized and death from botulism is basically due to respiratory failure that happens because of muscle paralysis of the respiratory organ. So it is the toxin is basically an AB type of a toxin. So let us look at its mode of action. So again, the wild type like we have the non-pathogenic strains of Clostridium botulinum, which are not producing the toxin. However, because of integration of a phage, so the lysogenized bacteriophage that is integrated within the Clostridium botulinum genome basically encodes for the toxin. So during what happens during the normal uh, presynaptic membrane, so there is excitation signal that is received by the motor neurons from the central nervous system, and that leads to release of uh, acetylcholine molecule. Uh, uh, so the acetylcholine molecule that is being released by the nerve cells will bind on the surface of the muscle cells. Okay, so there are receptors for acetylcholine on the muscle cells, and that will lead to muscle contraction. Now, so in this disease that is known as botulism, what happens is uh, the excitation signal is still there from the central nervous system, and in response the neuron is supposed to release acetylcholine. However, because of the presence of the toxin, it will bind to the neuron, and it will inhibit the release of acetylcholine. So as a result, the muscles will not contract. It will stay in relaxed conditions. And so basically, because of botulinum poisoning, the muscle is not receiving any excitation signal, and that leads to kind of a muscle paralysis of the respiratory tissues, leading to eventual death. Then the second type of toxin that we'll discuss, tetanus toxin. It is also a neurotoxin. It is also a EB type of a toxin. Now, on contact with the central nervous system, this toxin is transported through the motor neuron to the spinal cord, where it binds specifically to ganglioside lipids at the termini of the inhibitory interneurons. The inhibitory interneurons normally work by releasing an inhibitory neurotransmitter, which is amino acid glycine, that binds to the receptors on the motor neuron. So glycine from the inhibitory interneuron stops the release of acetylcholine by the motor neurons and inhibits muscle contraction, allowing relaxation of muscles. However, because of the presence of the tetanus toxin, it blocks the release of glycine. As a result, the motor neurons are not inhibited, and there is continuous secretion of acetylcholine that leads to uncontrolled contraction of the target muscles. And that leads to a type of paralysis, which is known as twitching paralysis, or basically the individuals will show certain spasms. So, this can, if a respiratory muscle is involved over here, then there will be prolonged contraction of the respiratory muscles, and that can lead to death. We also have a diagrammatic representation of how these uh, toxin acts. So, acetylcholine is secreted by the motor neurons, and to stop the secretion of acetylcholine, we have glycine molecules that are secreted by the inhibitory uh, interneurons. Okay, so when the glycine is released, then further release of acetylcholine is inhibited or stopped, and hence the muscle relaxation will take place. So this happens in a normal case. Now, because of the presence of toxin, what it does, it blocks the release of glycine, and as a result, uh, the glycine is not able to stop the or provide a signal of stopping the release of acetylcholine. As a result of which, acetylcholine is continuously released. That is understood by the muscle cells as a signal for contraction, and hence the muscle continuously contracts, and that could eventually lead to spasms and death. Then next we have the enterotoxins. So enterotoxins are those toxins that will affect the intestinal tissues, leading to vomiting and diarrhea. One of the common example is cholera toxin, which is encoded by the bacterium Vibrio cholerae that causes the disease, which is known as cholera. So the disease basically starts by ingestion of the bacterium in contaminated food or water. The organism will travel to the intestine where it colonizes and secretes the AB toxin. So so it is also basically an AB type of a toxin. So with the help of a B subunit, it can bind to the glycolipids that are found on the cytoplasmic membrane. And then the A subunit, once it has entered inside the cell, it will show activity, and it basically has an adenylase cyclase activity that will allow conversion of ATP into cyclic adenosine monophosphate. Now, cyclic adenosine monophosphate is an important regulatory molecule, and because of increased amount of cyclic adenosine monophosphate within the cells, that leads to induction of secretion of fluoride and bicarbonate ions from the epithelial cells into the intestinal lumen. So this change in the ions leads to secretion of large amount of water into the intestinal lumen, as a result of which there is uh, loss of large quantities of water or dehydration that happens in cholera. So the treatment is generally the oral fluid replacement, uh, such as electrolytes, are used to balance the dehydration. Then the third category of exotoxins are cytotoxins. So these have specific toxic action upon the cells and tissues or organs. For example, nephrotoxins will affect the kidneys, hepatotoxins will affect the liver, and cardiotoxin will affect the heart. So they have a specific mode of action as well. They basically are membrane disrupting exotoxins, and they can be divided into two subtypes. So membrane disrupting exotoxins mean they are basically going to affect the membrane or the plasma membrane. They can be divided into two subtypes. The first type is basically consisting of a protein that binds to cholesterol molecules on the plasma membrane, and it will degrade the cholesterols, leading to formation of a pore. This will lead to a leaky cytoplasmic membrane, and osmolarity of cells is higher. So this leads to influx of fluids from the outside, that is the environment, leading to swelling of the cells and lysis. Examples includes leukocidins that are toxins that will affect the WBCs, that are synthesized by pneumococci, streptococci, and staphylococcus. Another example are hemolysins that will target the red blood cells that are secreted by the Streptococcus pyogenes. The second category of membrane disrupting exotoxins will basically targets the phospholipids that are present in the membrane. So they are basically phospholipases. Examples include Clostridium perfringens that causes gangrene, and the toxin produced is known as a alpha toxin. So they will destroy all the local WBCs that are recruited at the site of inflammation. The fourth category of exotoxins are superantigens, and as the name suggests, superantigens means they will drastically provoke the immune response, and hence are known as superantigens. Example includes Staphylococcus exotoxin, which is involved in causing food poisoning. So it causes release of massive amounts of cytokines by the T cells, and it is an important signaling molecule, and over secretion leads to disruption of this regulation and has a toxic effect. Lastly, the second category of toxins are endotoxins. So, gram-negative bacteria basically have, we have already studied this, they basically have lipopolysaccharide in the outer membrane. So these lipopolysaccharide, this is the structure of a lipopolysaccharide. It contains the innermost portion, which is known as a lipid A. Outer to it are present the core polysaccharides, and outer to these are present the O antigens. So the lipid A part of the lipopolysaccharide can actually act like a toxin. So endotoxins are nothing but the lipid A portion of the lipopolysaccharide. So some of the properties of endotoxin includes that they are heat stable. They are toxic at very high doses, about milligrams per kilogram, and they are weakly immunogenic in nature. They are capable of producing general symptomatic effects such as fever, shock, blood coagulation, weakness, diarrhea, inflammation, and intestinal hemorrhage. The biological effect of endotoxins are indirect. They can activate blood clotting factor 12, which activates about four different humoral systems, such as coagulation, complement, fibrinolysis, and kininogen system. These are also pyrogenic in nature, means they will lead to generation of a fever, which is basically because of release of certain cytokines such as tumor necrosis factor. So that's it about bacterial toxins. Kindly like, share, and subscribe if you like the content, and to get direct update on uploaded videos.